Okay, so today we are going to discuss about the model of plasma membrane. So here the first model was about that name was sandwich model. That was sandwich model. In this model, they proposed that the cell membrane is composed of lipid bilayer. Cell membrane composed of lipid bilayer. Okay, and the lipid bilayer and the sandwich between the inner and the outer layer of protein means that they gave the name as the sandwich model because in the mid we have the plasma membrane was composed of the lipid bilayer, and the outer and downward side there was a covering of protein, so that was looking like a sandwich. So this basic structure of the sandwich model is also known as unit membrane. Unit membrane. Now, and it presents the cellular organelles. So after all, the modern technology revealed that the lipids bilayer is not a sandwiched between the two layers. Means they said that the lipid bilayer is not the one type of sandwich between the two lipids. Let me tell you, the sandwich is like this. We have one bread, then here we have different material like potato, tomato, and there. So then again we have bread close. So here both are the bread, so these act as the protein, while this act as the lipid bilayer. Okay, so according to modern technology has revealed that the lipid bilayer is not a sandwich. जो कि ये लिपिड की बायलेयर है ना, वो सैंडविच नहीं है। दो प्रोटीन की लेयर के बीच में जो लिपिड की लेयर है वो बायलेयर नहीं है। तो इस हिसाब से सैंडविच के मॉडल को आगे मॉडिफाई किया गया। दैट वाज मॉडिफाइड बाय फ्लूइड मोज़ाइक मॉडल। फ्लूइड मोज़ाइक मॉडल। Okay, so this fluid mosaic model basically that was given by Singer and Nicholson proposed this model in 1972. So in this model basically they we can say here in the fluid mosaic model, Singer and Nicholson in 1972 proposed that here in the mid between we have a fluid present, and that fluid is lipid and that can easily rotate. Okay, that have able to rotate and transfer or vibrate. So, and the proteins are present in the bilayer of phospholipid, not lipid, bilayer of phospholipid proteins were present. And let me tell you with the example. Here we have okay, so here as you can see in the mid between, here we have lipid membrane and fluid is present. So this fluid is a lipid. Okay, and this is what proteins. These all are proteins, or these mosaic is basically protein. Okay, so now this fluid help us to rotate, to vibrate, to move, or to translate any of object. So here we were having the embedded protein, that was integral protein, and it is also known as intrinsic protein. Okay, so in the fluid mosaic model, that was that this model is basically iceberg in the sea. iceberg in the sea. How iceberg in the sea, like iceberg here was denoted as a protein and sea was denoted as a lipid. So protein are present in the lipid. So that was named as iceberg in the sea. Okay, so here proteins are sometimes completely embedded, or sometimes these are partially embedded, sometimes here. So here now we are having two type of proteins. Two types of protein. Let's discuss over here. So the two types of proteins, first one was we have integral and second one was peripheral. So integral proteins are basically the embedded protein which are completely embedded. And peripheral proteins are basically the protein which are present here, the protein which are present on the surface, like here we have extracellular and this is the cytoplasm. So the protein which are present on the surface, so those are known as peripheral proteins, present on the surface. And the peripheral protein, the main work is performed as a receptors or enzymes. Receptors or enzymes. Okay, so these were two types. So now hopefully this is clear to you till now. Now here we are going to discuss some more about that. Draw. Okay, so here we have phospholipid. So this part is known as non-polar. Okay, and this is basically the lipophilic. As I told you, here lipid is present, so this is lipophilic. And this is what is known as hydrophilic. And this is hydrophilic. Sorry, this is hydrophilic, this is hydrophobic. Hydrophobic, we do not love the water, do not take the water molecules, and hydrophilic, loving the water. So this is, as the name is, lipoprotein, because lipid is present over here, and these does not allow the polar substance, does not allow polar substance such as water and different, because these are hydrophobic, water not attracting against the water, these are, so that's why they do not allow the polar molecules. While this is the head, and this is also known as phosphate head, and this is the tail. Okay, so in the phosphate head, that is basically hydrophilic, water loving, and allow the polar substances. So these were the two types, hydrophilic, hydrophobic. So the head is basically the hydrophilic, which is the water loving, and the tail is hydrophobic, which is against the water. This is the lipophilic and lipophobic. Now this is lipophilic, which do not allow the protein substance. Now here we have, if sometime we have polar plus non-polar. So these both substances are known as empipathic substance. Okay, empipathic substances. So here, basically here what we have, this continuous arrangement, like maybe you have seen like the leaves. Sometimes the leaves there is some किसी किसी पत्ते में मतलब थोड़ा सा कट आ जाता है, किसी में ज़्यादा, किसी में कहीं पर, किसी में कहीं पे. So that is basically the discontinuous arrangement. तो उसमें भी क्या है, discontinuous arrangement. ओके, और ये आइसबर्ग इंडस्ट्री कहलाते हैं. Now we are going, in the next video we are going to see the transport basically. Okay, so hopefully this is clear to you till that.